Hi, everyone. Welcome back to Unravelling T Cell Recognition Insights from Immunology and AI. My name is Hashem Shakwi, and I'm delighted to be hosting this webinar series where we explore emerging discoveries and technologies at the interface of T cell immunology, data science, and artificial intelligence. If you have a research story that would you like to share with our community, or if there is a particular speaker or topic or paper you would like us to cover in this series, please do get in touch. Updates about upcoming webinars, podcasts, and educational content are all announced through immunointelligence.co.uk and my LinkedIn page. Recordings of these webinars are also made available on Unravelling T Cell Recognition YouTube channel to enhance accessibility and inclusivity. If you enjoy this discussion, please consider subscribing to stay informed about future events. Today, I'm particularly pleased to be joined by Dr. Stephanie Gaglione from the Gladstone Institutes MIT, a lead author of a recent immunity paper, Scalable TCR synthesis and screening enable antigen reactivity mapping in Vitiligo. This work introduces powerful new approaches for reconstructing and screening thousands of T cell receptors at scale at low cost, addressing one of the major challenges in our field, which is linking T cell receptor sequences to their cognate antigens. Stephanie, thank you so much for joining us today. We are very delighted to have you with us, and the stage is yours.
All right. Thank you very much for having me here. Okay. It's always been an honor to be on this particular series. Okay, so just an opener, obviously this audience needs no introduction to this. T cell specificity refers to the ability of T cells to recognize specific antigens. Every T cell has a unique T cell receptor that enables it to recognize antigens, which is a critical piece of information that we need to understand for disease pathology, to design targeted therapeutics, and to induce antigen-specific tolerance. And more broadly, and I think the core topic of this particular group, being able to predict T cell specificity from sequence. And a large part of this field is motivated, I always add this for those, I don't think this audience needs it, but understanding why this matters, that ultimately we do want to be able to engineer antigen-specific cells. And that largely at the present moment, the vast majority of T cell therapies are focused on a limited number of targets. And my personal belief here, that limited number of targets is due to our lack of understanding of what specific antigens are targeted by T cells. And of course, we have the flip side. As an example, type 1 diabetes, that autoreactive T cells can target cells specifically, and that being able to selectively delete T cells or constructing tolerogenic strategies will be key for the next generation of immunotherapies. But of course, understanding and predicting and knowing what T cells recognize which antigens is challenging due to the complexity of TCR-antigen interactions. We have extraordinarily high combinatoric possibilities. The interactions between TCRs and PMHCs is relatively low affinity, and TCRs are very promiscuous. And so the result of this complexity is that we remain relatively limited by our ability to collect high-throughput interaction data, and that finding therapeutically relevant TCRs and antigens remains difficult. And I think this is a phenomenal review that some people in the audience here contributed to that really made an impression on me as a graduate student. And that's that there are relatively few available TCR antigen pair data points in literature. The vast majority don't have both TCR chains, most are viral origin, and the vast majority are 70% of known TCR antigen pairs or a group of about 100 antigens. And this is true if you go into VDJDB or IEDB or any databases. You don't find a large diversity of TCR antigen pairs for many HLA alleles with many potential options. And I think it's quite clear that data availability is a principal limitation to our ability to predict, construct maps of how TCRs recognize antigens. And so the reasoning from the perspective of a tech developer is that this is a tech problem. Antigen-directed methods exist to screen many TCRs against relatively few antigens. And so, of course, multimers, APC assays, things like ELISpots. Conversely, we have TCR-directed methods. So these screen few TCRs against many antigens. And of course, the challenge moving forward here is that we want approaches that can do many TCRs against many antigens, and to be able to do it, in my view, with features that make it amenable to broad adoption. So not just scale and specificity, but also speed, ease of implementation, low cost, applicability across MHCs, and then approaches that can fill that space are what are truly going to create large data sets, that it's not going to be a single group that's building the data that will enable predictive capabilities, but that it's the ability to have broad adoption of tools that will enable the types of data that we need in order to be able to predict. And so today I'm going to actually show two stories. I'm going to show, and then this data is presently published, but I'm going to hint at some new work that we're doing in Alex's lab here. But the first is being able to screen T cells from patient samples at scale. And the second is to be able to expand on some of these tools to do deep mapping of the TCR-antigen interface. And the first is close to my heart. So we were working quite closely here, and this is work that I did in Michael Birnbaum's lab, working quite closely with some collaborators at UMass Chan and Pfizer with a particular data set that they found to be important or rare enough that it could help us address the question of what specific antigens do self-reactive T cells target in vitiligo, and are there similarities between T cells responding to vitiligo and melanoma? And just as a brief background for this audience, vitiligo is a skin disorder caused by autoreactive T cells that are destroying melanocytes. And the general vision of the field here is that it would be ideal to induce immune tolerance, antigen specifically, to control disease without long-term generalized immunosuppression. And that's a challenging goal, but that the first step in it is being able to build a pipeline that will allow you to go from data that looks like this to maps of what TCRs are recognizing which antigens and what cell state they're in. So in this case, we had a data set from 10 of the HLA-A2 positive donors that had vitiligo. Suction blisters were generated on lesions in order to extract blister fluid, and that blister fluid contained T cells that were further processed not just to acquire the TCRs, but also to have matched transcriptomics. And we also had PBMC data from this as well. And so if you were to take this type of data set in a conventional, a single cell study, you would construct a map, you would look at the cell states, are they naive, tissue resident, Treg? But insights from the TCR sequences are relatively limited without the data on what antigen reactivity is. And that information provides color as to which T cells are potentially pathogenic and also which T cells are bystander. And if you're confronted with a similar type of data set, the typical approach here is to select a short list of TCRs. You would pick the most expanded, you would pick the ones that are exhausted or activated, cytotoxic, excuse me, whatever cell state we found to be the most important. And the reasoning, I think, is largely that current screening approaches are challenging and expensive to execute. And so for a similar type of question, you would probably pick a tool that allows you to screen many hundreds of TCRs against many antigens. But for any questions that are in this purple region, I didn't really have an approach to be able to look at it. And so we thought, let's try and build some tools that will allow us to fill that gap, or at least begin moving that direction. And I identified two problems here. The first, the TCRs are very expensive and difficult to reconstruct from sequences, and not accessibly. And two, that screening methods to pair TCRs and antigens scale poorly. And so let's take a look just at the first problem and what we decided to do with this one. So of course, TCRs consist of both highly variable and constant regions. Each has a constant chain, as well as a region of moderate variability defined by the alleles that each alpha and beta variable region are. And each TCR contains a unique CDR3 beta that defines ACT with the peptide and an MHC groove. And if you were to reconstruct, the two most popular approaches here are two. You can synthesize the TCR, the entire TCR as a gene fragment, for roughly $100. Or you can try to manually assemble individual TCRs by taking the CDR3s and using stocks of the different variable regions to reconstruct them. But this manual reconstruction will either require you to have some liquid handling capabilities, or an army of very willing grad students, and that neither of these was really viable if I want to reconstruct 3,800 TCRs. And so we had some discussion of what we felt were the right criteria for our new approach. Low cost, we said we want something less than $1, easy to execute, requiring only common molecular biology reagents that anyone can execute. You could put a pipette in someone's hand who's done only basic cloning, or maybe not even, and that they could execute this. Highly scalable and compatible with the large breadth of different assays that the field uses, not just the ones that we were using in Michael's group, but also other types of antigen screening assays, APCs, whatever. And so this is where we introduced TCRaFT. So TCRaFT is our approach for pooled assembly of TCRs and sequences, and it uses actually a relatively simple concept. So just at the baseline here, we realized that if we had both CDR3 alpha and beta ordered in a linked short oligo, that we could facilitate correct pairing of this oligo with a pool of vectors that contain 48 TRBV alleles. And essentially, each one of these oligos has to match to one of the 48 vectors. And then we take that resulting product and execute a second reaction. And the second reaction will then integrate one of the 45 alleles for the alpha side into the same library. And from here, we can then transfer this complete product an expression vector of choice. The key distinction here is that we are doing this in pool. Now, the mechanics behind it and just how we thought about it, if you're trying to put both CDR3s just by the average lengths of human CDR3s into vectors, then you have very little space to allow for any type of complementary sequences that would let you pair these oligos with vectors. And really the only viable option we had realized was going to be Golden Gate. Golden Gate, for those who don't spend a lot of time doing cloning, is a very well-precedented approach where you're using type 2 enzymes to cleave outside recognition sequences that then reveal 4-base overhangs for you to facilitate pairing using complementary overhangs. Now, this is typically used in the context of trying to link many sequences in series. And we had the thought of what if instead of doing series, we did this in parallel? And NEB had just released, and not too long ago, a couple of years ago, a data set of all possible 4-base overhangs against all possible 4-base overhangs. And that data set could then be optimal for trying to construct orthogonal sets of 4-base overhangs to facilitate proper pairing of different components. And so what we did here is we designed overhangs that would then, for each CDR3, we had a first reaction where you had sets of overhangs on the outside that facilitate pairing with the first set. So this is not unique to each CDR3, but it's unique to each vector. So for each of the 48 vectors, there's 2 4-base overhangs. Same for the second step, which then results in insertion on the inside. And the little trick that we realized when we did the first experiments and said, oh, this is definitely going to work, is actually that in series-based Golden Gate reactions, a single mispairing event results in an incorrect product. If I were to try to link 15 things in series and one mispairing happened between 2 of my overhangs, I wouldn't end up with the product that I want, but I would potentially end up with something that transforms, that produces a product nonetheless. But because we're using 2 overhangs, a single mispairing event produces something that doesn't transform. And additionally, if something does happen where you have a mispairing at some point in here, you also end up with a product that's not a TCR. And so there are some safeguards within this type of system that we realized just very elegantly ensured that you can relatively easily filter out anything that would produce something that was not a productive TCR. And we initially had set a goal internally for ourselves that if we could get something like 70% of the TCRs to be correctly assembled, then we were going to be happy. But we ended up with something that was a little better than that. So here, what I'm showing on the y-axis are all of the alpha and all of the beta alleles, and on the x-axis, the 3,800 TCRs. And we were showing here red that we had correct pairing and gray incorrect pairing. And I remember bringing this to Michael, and Michael goes, well, I have to squint to see the gray, so this has to be pretty good. And we had 99% of the TCRs detected, but also that the distribution was reasonable enough for us to go ahead and use it as a library. We have no PCR amplification steps past that first oligo amplification, which really helped with producing libraries that are consistent with what you start with, as the oligo distribution is more or less what you're ending with in a normal assembly. And so from here, just to emphasize why we felt this was something I know was quite useful, that this first library cost us $5,700 to build, and versus a little cheeky here, but at spending $100 per receptor, we weren't going to be able to do this with standard synthesis. So that takes us to the second problem now. You've got a TCR library. How do we screen it to pair TCRs against antigens? And so from here, I'm going to just recap the strategy that we were using in Michael's lab to be able to infect cells specifically. And so typically lentiviruses have a fusogen, VSVG, that targets cells via the LDL receptor. And in 2018, there's an identified structure that enabled you to pinpoint specific mutations that would eliminate the binding function of VSVG without abrogating the ability for it to function as a fusogen. And so then you can replace the binding function with a targeting domain of interest. And in this case, that is a peptide MHC. And so what can one do with a peptide MHC displaying lentivirus that has a modified fusogen? You can construct libraries of these PMHC displaying viruses that also encode in their genome a barcode for what peptide is being displayed, and mix it with a TCR library that PMHC viruses infect cells through the TCR, and you can then sequence the cells to reveal TCR-antigen pairs. This initial approach was demonstrated in a proof of concept by Connor Dobson back in 2022 on a Nature Methods piece that has been quite impactful. And from here we said, all right, let us use this for the first time. Let's apply it and integrate it with this TCR library that we've constructed. See if we can pull out the TCR antigen pairs in a true first library on library actual data set that we want to examine to look for reactivities. And so we constructed, to start here, a list of 101 peptide MHCs, and that this peptide MHC list was arrived as melanoma related. We pulled out anything from the literature that we felt to be an important melanoma or vitiligo antigen. We then constructed the library, did this approach that I just described, Raptor, used the viruses to infect our 3,808 TCR library, and then sorted and sequenced the pairs. We identified that there's one little key thing that really did help with any little bit of nonspecific transduction, and that is at a very early time point to use activation as a means of selection initially. And so we know that these PMHC lentiviruses are potent activators of our expressing cells. And then if we do that type of initial sort, we can, just from bulk data alone, so this is just bulk data, fold enrichment of TCRs over the baseline frequency, and here just the proportion of reads for peptides that were in the library, we can pull out which ones are reactive, not necessarily paired, but just bulk data, which TCRs are coming out as activated by the PMHC library. And we can then do 10x to take the cells and from there pull out which peptide MHCs were linked to each TCR. And the way that we did this is by looking at assigning each cell a peptide MHC identity. So looking at the number of UMIs for a given peptide associated with a cell barcode to construct plot of which peptide MHC is associated with each TCR. So as an example here, for this particular TCR, it's clearly reactive to this MART1 epitope. And here for this TCR, it's reacted to two related GP100 epitopes. And you can do this strategy across clones that come out of the sequencing. So for example, here on the y-axis, we have TCRs. On the x-axis, we have antigens. I've grouped some on the left here. If we take a slightly closer look, each row is essentially one of those bar plots that I just showed. So for TCR 2799, this recognizes MART1 2635 epitope, the sort of conventional EAA. And we can do this across different peptide antigen pairs in order to identify which specific antigens are recognized by TCRs. And from here, of course, this work was led by Chirag at Pfizer. We can link this reactivity data back to the transcriptomic data. And we had also, not showing it here, but of course it's in the paper, we had also then orthogonally used APC-based T2 cells pulsed with peptides, some larger peptide pools, to do two things. One, demonstrate that our TCRaFT-based approach is very compatible naturally with other antigen screening assays, but also as a means of validating that we were pulling out correct hits. And so in total, we identified 138 TCRs that were melanocyte reactive, 24 MAR1 reactive, and we also took a look at what the top undetected TCRs were. And we pulled out some common features just to highlight what we were looking for and to demonstrate the broad utility. We can look at if you can reconstruct every TCR, you are capable of looking at clones that are not clonally expanded and identifying the dynamics between different types of reactivities. You can also look at the enrichment of specific alleles. Of course, MART1 reactive TCRs tend to be TRAV12-2 as their alpha allele, which aligned well with the data that we pulled out from these particular samples. And we also were able to link this back to some wonderful published signatures on melanoma from Kathy Wu's group. And then we just want to show that this really does scale, that there's no limit because we're using overhangs that are on the vector side and not on the oligo. That's what enables this to be, there's no change in the complexity of a reaction if you had 1,000,000 TCRs or if you had 100. And so effectively that reaction is the same. And so from here, we, in partnership with Stephanie Gaglione's group, reconstructed 30,800 TCRs in less than a week from 21 PDAC donors. And that, of course, again, a little cheeky here, but this really just wouldn't have been viable with any other approach so cheaply, inexpensively, and easily. And that we were excited that we could take this list, quickly build it, and then demonstrate, again, the same result, pretty consistent because, of course, we're using the exact same vector pools. And from the perspective of the reaction, there's no difference. But that we could also then take this and use it in the context of initially here, we just did T2 cells, pulse it with peptides, and pretty sensitively pull out clones that are reactive to specific antigens. And there's ongoing work here to deconvolute specificities beyond the 30,000, beyond the common viral antigens, but that was not the focus of this particular piece of work. And so just to summarize this particular work, here what I've shown you today, and then the last bit will be about 10 to 15 minutes. This first project is showing our ability to take sample single-cell sequencing, reconstruct thousands of TCRs, and then screen those TCRs and antigens in pool to capture reactivity information. I'm just incredibly excited for what potential this is going to bring as far as our ability to look at important biological questions that are driven by knowing what antigens are recognized in the context, especially of tissue and particularly in autoimmunity and oncology. All right, and then the second, just wanted to briefly link this to some work that's a little more fundamental. And this is to map the TCR-antigen interface, where if you want to be able to tune that specificity and screen millions of TCRs, that we need some different capabilities. And so in the context of Raptor, viruses bind and infect cells with corresponding TCRs, and in a very useful feature, it's activation-based. But this particular strategy of building these peptide MHC displaying viruses is also applicable if you want to construct VLPs. You can display the peptide MHC on the surface. There's barcoded RNA in the VLP that can indicate what PMHC is on the surface. We can also make them fluorescent, building on some work that others have shown. And that these are generated at library scale by making a cell line that you can then very easily reconstruct or very easily generate particles or VLPs. And so we thought, can we use VLPs as a reagent to detect binding? And can we do it in a context where we can screen easier and more broadly at extremely high throughput? And so I'm going to just briefly show yeast display. Yeast display is very highly precedented, of course, for having 10 to the 8 plus options on the yeast side. So you can have TCRs, you can have peptide MHCs, and this is work that was really led by a large, spearheaded by Michael while he was in Chris Garcia's lab, as far as the peptide MHC side, being able to look at TCR cross-reactivity. And the typical way you do this is by generating recombinantly produced protein for your PMHC or TCR, and then selecting and sequencing binders. And you can do this multiple times for different libraries, different multimerized PMHCs or TCRs, but inevitably you're going to run into the limitation of needing to produce recombinant protein and needing to execute these screens in array. And we had the thought of what if we could replace the recombinant soluble PMHC with VLPs that would allow you to multiplex screening, enable mapping, and also set the stage for non-soluble, not needing to use soluble protein in order to execute screens. And so just as our proof of concept, we created some TAX-A2 VLPs and some SL9A2 VLPs. ungenerated yeast that had single chain TCRs, which is the current optimal way at the moment, at least publicly, for expressing TCRs on yeast. And we demonstrated briefly here that they were able to bind their corresponding TCR. So here we have the y-axis being flag expression that corresponds to TCR. And on the x-axis, we demonstrated also that you could do secondary stains with beta-2M to amplify and boost the signal of which VLPs are binding, and that they match. And of course, though, we want to do this at library scale. So we generated the theory here is let's generate a library of A6 TCR variants and a PMHC displaying VLP library and execute a sort of similar workflow to what we were doing in the context of Raptor, but with some significant changes. The first is that we are using yeast in significantly larger libraries, which I'll about to show. And the second is that we're trying to sort and sequence yeast, which ultimately is not very compatible, as it turns out, with 10x. So as our first proof of concept, we took this A6 TCR, we randomized it at 3 positions that contact the peptide, which generates about 7,000 unique TCR variants. And then we constructed a sample library where we have an on-target PMHC VLP and 96 off-target PMHC VLPs. And then you do some rounds of enrichment, wash away what doesn't bind, and then do a quick RT-PCR and NGS for the VLPs that bound to the enriched library. And this was the moment I said, all right, this is a pretty clean platform. We don't see much in the way of off-target VLPs binding. So this is just unprocessed read data. There's no processing. This is simply just pulling off what of the 97 VLPs bound the A6 enriched yeast. And we further then said, all right, let's increase the scale of this and let's try to do some mutation on both sides. So if you were to try to do this with conventional approaches, you would have your mutagenized yeast library, and you would also then, for every peptide that you generated, generate it recombinantly, execute a screen, and try to build a map that way. But instead, we decided we're going to use our PMHC library-based approach as our proof of concept, and generated a library with a diversity of 47 million variants against 92 PMHCs, including 37 that are randomized at 2 positions, here 5 and 8. And at this scale, you really can't do fluorescent-based sorting. This is something that's just not viable. And so we identified actually that our PMHC VLPs, been using beta-2M, it was compatible with doing MAX. And so what this enables us to do is screen billions of yeast simultaneously against the 92 VLPs, where instead you would have had to do 92 separate selections. And just to show what the bulk data looked like, here, same process. X-axis is the composition of our library. Y-axis is just the frequency of the reads, the proportion of reads. Here we have the wild-type tax peptide. What you can just see at the bulk level of the VLP library is that mutations at position 8 are not tolerated very well in the tax peptide as far as it impacts binding across variants, even mutagenizing completely those positions. And we executed the same, not going to show much of the data today, but we did the same thing for SL9 and the 868 TCR. But of course, in order for this to be something that's really broadly adoptable or useful, we need to be able to pair the TCR-antigen data. And we had some phenomenal collaborators that had an approach we identified to be very complementary for this particular use. I had tried doing 10x, didn't work out very well. And also, you can only do that tens of thousands of cells at a time and very expensively. And Francis Keir and Paul Blaney's group has developed, and this was just posted on bioRxiv not too long ago now, an approach called Stitch-seq. And Stitch-seq enables you, without any kind of bead capture, to do overlap extension to link sequences together. And she had initially envisioned this in the context of TCRs and antigens, but we identified this could be exactly the tool that we need. And it worked really out of the gate. So we encapsulate the yeast. After enriching them, we can take millions, in this case, we just shoved 5 million in, of our VLP-stained yeast. And from here, we stitch together the alpha to the peptide and the beta to the peptide. And we also had bulk sequenced the yeast alpha beta in order to pull out what those pairs were from our amplicons. But theoretically, we could have stitched, depending on how we constructed this, slightly differently. But this is just to ensure that we could do short read sequencing to pull out the pairs. And then from here, we can reconstruct alpha beta peptide pairs that allow us to build a map of, from this enriched data, what you can do. So from this enriched data, what alpha, beta, what TCR antigen pairs existed. And so here's just the map. I've highlighted the wild type peptide here. And really at this stage, we actually have just constructed this data and done some validation, which I'll talk about momentarily, but we haven't really taken it farther than this. And this is just our sort of initial point, demonstrating really that there is some sensitivity to how very small alterations in a TCR sequence can alter the peptide binding quite sensitively. And just as an example here, showing the difference between wild type and Q at position 5, that there are some very significant differences in which particular residues are tolerated at each position. And really at this point, we just were looking to build a type of deep data set that wouldn't have been possible without an approach that allows you to multiplex at this particular scale. And so now just to do a quick recap before questions, the general importance of this work, we collectively in this particular audience know how critical it is that we both understand and have tools to identify T cells and antigens driving anti-tumor responses and autoimmunity, and that our eventual goal is to be able to build targeted therapies and understand disease in a way that we presently don't. And that today I've shown two pieces of work. The first, that we can reconstruct TCRs from sequences in a broadly adoptable way. I think we've had quite a few groups adopting it now, which has been really wonderful to see. And it's on Adgene if you haven't tried it out yet. And you're always welcome to reach out if you have any issues or problems adopting it. And that we can integrate this with approaches that are still ongoing. I think we're at such an exciting moment now to map them to antigens in pool. And the second is to do deep mapping of the TCR-antigen interface by using these type of strategies that allow you to build scalable libraries, but bringing them to a realm where mammalian cells offer a very distinctive advantage, but being able to identify some of those strategic strengths of yeast display is what will also enable us to do really deep TCR mutagenesis and screening, and hopefully design, at this point, some phenomenal screens that will build the type of data that's has higher resolution on how TCRs are interacting with antigens. And I just want to, before some questions now, want to do an incredible thanks to the people that were instrumental in building this. And most of this was my graduate work with Michael Birnbaum, and that I owe some phenomenal credit. It's a really high credit to a couple of people. And I especially want to highlight Rack, who is a co-developer of TCRaFT, and Chirag, who led our analysis of the integrating data for vitiligo and melanoma and being able to pull out some of those insights from gene expression. And my new group here at the Gladstone Institutes that has taken on the mantle of some of this work, and that we've integrated and pushed the boundaries on the antigen side into new territory. And of course, I came from Oxford before this, and so I love staying in touch with the Oxford community. And I'll be in town, actually. I'm flying over this week. So if anyone's over there and up for meeting, I would love to chat. And so with that, I'm very happy to take some questions. And I really appreciate everyone being here today. And I really do love the community. I feel like we're at such a moment where everyone knows everyone, and we're all working towards the same goals. So I'm very grateful to be part of that community. Right. Thank you.
Thank you so much, Stephanie. Brilliant work, very fascinating. I enjoyed from the beginning to end, really.
Thank you.
Thank you. So we have a good amount of time for questions, please. So maybe I'll start. I have actually a couple of questions very related, but they are related to first part of your seminar. The first question is that obviously the peptide MHC pseudotype lentivirus screen is elegant, but what we know is that TCR peptide MHC affinities span several orders of magnitude.
Yeah.
Some autoreactive TCR bind very low affinity. So my question is, how did you calibrate and set the threshold?
Yes, so that's a great question. It's something I think about with a lot of new approaches. The first is, we did do testing across the physiological range of affinities. That was pretty important. So pulling out, this is actually from work done in Omer's lab, sets of TCRs that had validated antigen affinities from using SPR, and then being able to confirm that we could hit the low, like 100 micromolar without an issue, even as low as 150, being able to detect it. But of course, I didn't really feel that was enough. And so we had decided to, in the context of this work, to use peptide pulsed APCs for that reason, that we pulled out, and this is in the paper. I don't know if I put it quickly and accessibly in my supplement here, but being able to show that we could pull out the exact same TCRs using an approach that has worked pretty universally for even low affinity antigens, being able to capture that. I will put the caveat that we were doing this in jerkats, and jerkats do have some lower sensitivity, and that's something that I'm sensitive to. I think Giacomo Oliveira on Kathy's group, I think it was Giacomo, had a paper showing that there could be some TCRs that in jerkats don't have quite the same sensitivity to antigen that they do if they're in primary cells. Cognizant of that, every tool has its pros and cons. So that's the way I look at it. But no, to answer briefly, yes, being able to capture low affinity antigens is important. And then I think we've done enough profiling to feel confident that we're pulling out low affinity TCRs as well. Whether it's all of them, still, as time will tell. Some good questions.
Yeah, before I move to questions from audience, if I may, I would like to ask the second question because it's very related to the first. And my question is that if my understanding from your first part and your paper is correct, a good fraction of more than 3,800 TCRs that you scanned, they didn't show any antigen specificity to any of the antigens you tried. So the question is it a limit of the technique, or is it a question of how to put the threshold? And also, can those non-binding TCRs or non-binding data can be considered as a negative data in modeling, because lack of negative data is a major problem in limiting the modeling?
Yes, a great question. A few things. One, we only did A2 here. Two, we only did 101 antigens here. So I think at the baseline, we did cover a lot of the most important ones. So we're certainly missing a lot of reactivities here. And we're doing one of the many, this is 10 donors, one of the many alleles that could be presenting peptides here. And so this is some work that I'm thinking about very actively, that in order to really be able to deorphanize many TCRs, I do think you need to be in the tens of thousands at this range of antigens in order to capture reactivities. That said, because we did do multiple approaches here with the exact same peptides and the exact same TCRs multiple times, the negative data is actually relatively strong. That we, and Rack, here now a PhD student, has taken a bit of a look at this. The negative data does seem to hold true, that we have demonstrated if it's not reactive to the virus and it's not reactive to a T2 post with the same peptide, it's probably not reactive to that antigen. So we feel fairly good about negatives, but I do think that we can take this library and pin it against a more expansive set of potential antigens to be able to deorphanize. And I think the low deorphanization rate really just highlights how challenging this type of work really is. Yeah.
Sure, thank you. The next question is from Robert Sparpan. Thanks for your inspiring talk. You mentioned the importance of JARCA T cell activation in NAPTR. Have you ever seen differences in capacity of certain TCRs to activate JARCAs as deactivated by CD69 versus NFAT reporter? And if so, which pathway of activation do you think is more relevant to show functionality of a given TCR? It's a great question, actually.
Interesting. I think jerkats are inherently limited just in and of themselves. That is my view. And so I think that as far as showing functionality of a TCR, whether it's via CD69 or NFAT, the real reason we used NFAT here, having tested both, is that NFAT is more sensitive, just much more sensitive, having that fluorescent reporter. There does seem to be some baseline CD69 in jerkats that you can get enrichment, very nice enrichment, but if you really want that extreme sensitivity, the NFAT reporter really does do a phenomenal job. Now, also, you did look at correlation between NFAT and CD69, not with the viruses, but with the exact same set of peptides in T2 peptide pulsed APCs. And it pretty much has a correlation. We did see that CD69, of course, has some more background noise, but anything NFAT positive was CD69 positive. I think functionality inevitably can be shown via cytotoxicity in primary cells, but that here, an NFAT reporter is from the literature, a pretty good way to get high sensitivity readouts, regardless of what the antigen stimulus is in jerkats. I see the first question from Sharon, and yes, they're all published. I just put the two ad gene links in the chat. We put them up as both options for, you can order all of the vectors in array and make pools yourself if you want, or use them individually if you should desire to do that, and also already pre-pooled. And so we put sort of everything there with worksheets. Yeah, everything is there. And if you have any issues and you contacted Adgene, they're going to send you to me. So I'm happy to help if you have any questions or issues with it. Velociraptor, yes, up as a preprint, but not quite in exactly the same depth. But yeah, always happy to chat about that. And then Ilya, yes, we only orphanized 5% of the TCRs. Not much to argue with that one. Unfortunately, I think that you are correct here. And autoimmunity is especially difficult. I think that was part of the reason we picked that as our first application here. But I do think PTMs are, of course, incredibly important in autoimmunity. And so we're actively thinking about ways, I'm actively thinking about ways too as a postdoc of how to begin tackling that. I would love to chat with anyone, if anyone in here knows a lot about PTMs in the context of TCR recognition and maybe brainstorm on that. But I think in conversations I've had with Michael too, that scalable screening of PTMs is going to be challenging just in generating cell lines that can produce certain PTMs reliably, that we can read out what those PTMs are. Some of them might just be hard to encode. But yeah, I think that is definitely a limitation of a lot of these approaches. Thank you.
Yeah, I don't see any more questions. Just to thank you. And also, for those who don't know, I have actually the pleasure of having Stephanie on board for a podcast discussing a very similar topic. And actually, Stephanie, one comment I got from the podcast was that Stephanie is not only a great scientist, but also very fantastic storyteller. So thank you for that, to taking us through those technical terminologies and difficulties, but in a language that is accessible to a broad audience. So thank you so much for sharing your work with us. I truly enjoyed, and yeah, looking forward to seeing you next week, hopefully.
Yes, thank you so much, everybody. And yeah, glad to be in this community.
Yeah, okay, thank you. Bye-bye. Bye-bye, everyone. Thank you for listening. Thank you.